Hello, so today I wanted to talk about the anatomy of the nucleus. I thought that this would be important material to better understand the application of transcriptomics, like RNA-seq or PCR is one dimension of understanding the cell's function. So putting it into the larger context of what's actually going on in the cell is important for a higher level of understanding. And I'll be talking about some things I found interesting in this free book from OSU, and I've also sprinkled in some papers to make it more interesting. So the structure of the interphase nucleus is quite complex. On the outside, there is the endoplasmic reticulum, and this kind of hugs the nucleus, and it's studded usually with ribosomes, which, as you should recall, are the site of converting mRNA into their amino acids. And what is quite interesting about this relationship is that the outside of the nucleus is the ER, but the inside is also facing the nucleoplasm or the DNA, and they're kind of combined. It's two membranes. So what this does is optimizes and facilitates the moving of mRNAs from the nucleus with all the genetic material to the outside endoplasmic reticulum where all the protein production machinery is. So that is just one of the ways that higher level life has really optimized itself. Furthermore, you can see that there's a lot of other structures here that I'll talk about. So the cell you see in the textbook is kind of inaccurate. It's not really proportionally accurate to what you would see under an electron microscope. For instance, the endoplasmic reticulum should stretch way further out than how it is in here. If you think about it, it would be kind of inefficient to have such a little endoplasmic reticulum. Also, mitochondria could be bigger, Golgi could be bigger or located elsewhere. It's just very simplistic. So whenever you're thinking about the cell, you should think more about the transmission electron microscopy images you can see on the right here. It's a lot more accurate. It's what's actually going on. But the DNA has also organization too. You see a lot of the times in textbooks where it's just kind of this ambiguous spaghetti, but there's such a high level of organization, which we will get into. So, starting on the outside of the membrane, there's this really interesting component called the nuclear lamina, and it kind of hugs against this inner membrane here. And it, as you can see, it's kind of this meshwork of proteins that helps support and shape the nucleus. And you can have these transmembrane proteins embed themselves into the lamina and go through the double membrane system and connect onto the cytoskeleton. This allows for the nucleus to kind of position itself centrally in the cell. The position of the nucleus, as you can imagine, is essential to its efficiency or the optimization. You don't want it to take too long to get from one end of the cell to the other. So if it's in the center of the cell, it's the most optimal position to do molecular biology in. And also, it helps hold the nucleus together and position it. So there are also laminin-associated domains, which allow chromatin to anchor onto the nucleus. Now, these are very important in mitosis, as we'll get into, but the attachment points are on centromeres and telomeres or other structural regions of DNA that if you attach some protein to it, it's not going to interfere with critical gene expression. These region should usually be in heterochromatin because it's less likely to have any expression going on there. If you don't know what heterochromatin is, it's DNA that is more tightly wrapped around these proteins called histones. They're condensed. There could either be no genes there, or those genes aren't essential to the cell at that current moment. So what you really need to understand is that this lamina forms direct connections to the cytoplasm outside of the nucleus by anchoring the outside to the inside, and also that transcription factors can bind to the lamina as a form of repression. Signaling proteins can get sequestered into the nucleus and use the lamina to actually polymerize and be excreted through nuclear pores in that regard. But in mitosis, lamina are phosphorylated, causing them to undergo a conformational change to disintegrate the entire nuclear membrane. So breaking down of this nuclear lamina network will tear apart the nuclear envelope and cause this total structural disintegration, but it's still attached to the DNA because of those LADs. As you can see here, the phosphorylated laminin is attached to the membrane, some attached to the DNA in prophase. So you can imagine whenever the chromatin lines up during metaphase and is ripped apart during anaphase, all you have to do in the new cell is take off those phosphates, and it's energetically favorable to put the structure back together, as you can see in the sister cells. So it's really quite easy to rebuild the nucleus. And that's what happens after mitosis. This is done or controlled by CDK1, which activates kinases such as PKC, Aurora A, PLK1, VRK1 to actually do the phosphorylation of these lamina. As you may know, Aurora A, PKC are drug targets in cancer. Some antimitotic drugs target these kinases to prevent nuclear breakdown, which then prevents mitosis. These are your taxanes like paclitaxel, docetaxel, and other alkaloid drugs. So this is how they're working. Also, in cancer, the entire morphology of the nucleus is a little bit more sensitive. It's a lot more fragile. There are invaginations, multilobulations, distortion in the whole structure of the nuclei, and a lot of fluctuations in the actual nucleolus or other nuclear bodies. And clinically, doctors or pathologists can look at the nucleus and its structural morphology to determine the degree of a cancer malignancy and assess your prognosis. If it's a lot more weird or aberrant, then the further stage your cancer is in. And especially in cancer such as breast cancer, it's very fragile and lobulated, and it kind of has a hard time withstanding pressures in the cytoskeleton or from chromatin-based forces like in mitosis. So the actual cancer cell is, although very aggressive in proliferation, it's not as robust as the normal cell in surviving. So in cancer, this isn't a cause of cancer, it's more so a symptom of cancer, and the drug can exploit this fragility to kind of blow up the nucleus, thereby killing the cell. Furthermore, in the organization of the chromosomes in the nucleus, there is kind of a hierarchy. So you can imagine the whole cell as this solar system of different organelles revolving around the functions of the nucleus. And the nucleus is kind of like Earth, where if you look at it, there's different territories or countries. And those countries have different cities or states. And it's whole tree of order. So in the nucleus, gene expression is actually organized in the territories, where different territories use expression to promote different genes, but they all have functional similarities, as you can imagine. So chromosomes are maintained in discrete territories, where transcribing regions tend to be closer to the center or the nucleolus, and inactive domains that are gene poor are more so on the periphery of the nucleus, again, to anchor into the actual nuclear structure. And topologically associated domains are where the loops, or as you can imagine, where the trombone model happens, and these ensure crosstalk between enhancers and promotes facilitating co-expression. Within a topological domain, there are multiple loops, and these are just microenvironments of enhancer-promoter interactions. So going further with chromosome territory, A compartments are gene-rich and actively transcribed. They're really in the interior of the nucleus and interact with other chromatin regions. And these are where you'll find your topologically associated domains. B compartments are gene poor and transcriptionally inactive, again found near the periphery, and they interact with other inactive chromatin regions, and these are connecting to your lamin. Now, there's some confusion whether A-B compartments are the same as euchromatin or heterochromatin. For the most part, an A compartment is euchromatin and a B compartment is heterochromatin, but the A-B compartment is the big picture of that region. Euchromatin and heterochromatin are describing the local state of DNA accessibility. So you wouldn't find a topologically associated domain in heterochromatin because everything's condensed together, for instance, but a topologically associated domain can be between regions of heterochromatin. Furthermore, on an electron microscope, you can see regions of euchromatin and heterochromatin and distinguish them by their density. The less dense regions are more white, so you can see euchromatin is less dense, more open, more transcription is occurring, whereas heterochromatin is more condensed and less transcription is occurring there. And in heterochromatin, there are constitutive parts such as centromeres and telomeres, which are never transcribed and they're purely structural. However, there's also facultative heterochromatin, which are genes that are silent because in developmental biology they all start from the same cell type called a stem cell and then differentiate, and some cells don't need that. You don't want your eye cell doing things that your liver cell does and vice versa, so they specialize through epigenetic silencing. So just briefly for some nuclear bodies, these are thought to be involved in specialized nuclear functions. There are nuclear speckles in interchromatin regions that are enriched in pre-mRNA splicing factors, and euchromatin will tend to border around these speckles because these will act as a warehouse for SNRNPS, SNRPs, and SR proteins and have transcription factors and other things. So they're basically storing the splicing machinery. It's not where splicing takes place. Again, euchromatin will kind of be bordering these. You want to have a central warehouse so you can find everything and be able to ship it very closely. It's more efficient that way. And there's also Cahill bodies. Cahill is Spanish, it's not Cahill. I was corrected on that a couple days ago. And these are the site of initial modification of U-SNRPs. U-SNRPs are also in the spliceosome complex. So really what these do are contributing to histone mRNA processing and telomere maintenance, but primarily these are hubs to concentrate the enzymes for RNA processing. So there's also PML bodies, and these also are in the intrachromosomal space and cluster with other bodies, but they'll help with simulation and tag other proteins. But really what they are kind of thought to be storage molecules for having like a whole cascade of proteins ready to go. All you need to do is kind of throw your phosphorylate or throw your kinase in there to activate, say, a whole p53 response. As indicated in other studies, these will go to regions where DNA damage occurs and kind of sit there, and it's thought that, hey, the whole DNA damage proteins, or the proteins necessary for homologous recombination, will sit in these and then be shot out when they're needed. So again, it's an efficiency thing. So p53 modifying enzymes like CBP, HDM2, whatever, can be acylated or post-transcriptionally modified together to enhance the efficiency of this function. And also these can concentrate proteosomes and ubiquitin for easy degradation in the nucleus. It just really enhances post-translational modifications and makes things a lot more efficient. So the biggest structure everyone knows is the nucleolus. It's the most prominent nuclear subcomponent, where its purpose is to synthesize all of the ribosomes the cell needs to function. It's not really enclosed in a membrane. There's no membrane. It's just another density thing, because you have a lot of transcription factors and DNAs and other factors and stuff just sitting in here, because the cell needs to keep making a lot of ribosomes because it needs to make a lot of protein to do its function. So you have nucleolus organizing regions of DNA that actually code for ribosomal RNA, all folded into this region where factors and other things can express them and increase the rate of rRNA transcription. Also, interestingly, in terms of molecular biology, if you go to the regions of DNA that code for ribosomal RNA, they exist as tandem repeats, because you can increase your rate of transcription as that. If you have multiple copies, it'll be a longer transcript, but it can be broken up and have, say, if you have 3 repeats, you can get 3 transcripts for one transcription cycle. So it's very efficient and increases the rate of rRNA production. Also, transcription of different RNAs is mostly done by RNA polymerase 1, but the 5S RNA in the 60S subunit occurs by RNA polymerase 3. And for the actual proteins for the ribosome, it's done by RNA polymerase 2. So the actual nucleolus has subregions. So it has a fibrillar center where rRNA transcription occurs. Then on the border of this, in the dense fibrillar component, is where the pre-rRNA processing occurs. These are kind of like SNRNPs, but these are SNO RNPS. They're just the ribosome version of pre-RNA processing. And in the granular component is where everything is assembled. And then ribosomes are exported to the cytosol. And what you can actually see under the microscope is that the size of the nucleolus is proportional to the amount of metabolic activity occurring in the cell, and the granular component will increase or decrease depending on that. Also, for the nucleolus, there are caps that can form on the nucleolus in response to cell stress or transcriptional inhibition, and this is a survival mechanism. They are composed of reorganized nuclear components when RNA polymerase 1 is suppressed or harmed, and the caps are these temporary storage sites for nuclear proteins and RNA to help manage the cell stress and regulate the ribosome biogenesis. And they'll also happen when the nucleolus is undergoing DNA repair mechanisms to kind of preserve one of the main functions, the rRNA production for ribosomes. Now, getting into topologically associated domains, these are really used to constrain regulatory interactions between cis-regulatory elements and promoters. So a cis-regulatory element is like your enhancer or repressor, upstream or downstream, wherever in your DNA, that will fold over to meet your promoter with the mediator complex and initiate transcription. So this just keeps everything in line. So the right enhancer activates the right promoter. Chromosomes will fold into these domains with a preferential intra-domain interactions be indicative of these domains, and these genes tend to be co-regulated if they're in the same topological associated domain. What that means is that you'll have multiple loops in the same segment, and they could be in the same territory, so they're co-regulated. These could be perhaps the same pathway or doing sort of the same functions, so they're co-regulated or correlated with each other. Constraining the functional contact to a topologically associated domain, again, is essential for proper gene regulation. You can have large TADs, which are 880 kilobase domains, and they make up most of the genome. However, smaller 185 kilobase domains or subTADs are associated with specific chromatin marks. CDCF factors and SMC cohesion complex also exist at TAD boundaries to make sure that the transcription machinery kind of doesn't run off. You want to keep everything concentrated to your topologically associated domain or your loops. Furthermore, you can have super enhancers, which are clusters of enhancers that cooperatively assemble in a high density of transcription factors and coactivators to drive a lot of gene expression. And these form discrete nuclear condensates. There's so much stuff here, the properties of the solvent are kind of changing. And these are intrinsically disordered regions, and you kind of need this phase separation to enable this compartmentalization of all of these different factors and products. So these super enhancers are really essential for the cell identity gene regulation, like a liver cell making its digestive enzymes. You need a lot of it because it's a liver, that's just what it does, and it may cause cancer-associated dysregulation if, say, another gene got shot in here that wasn't supposed to be doing that, and it would unfortunately make a lot of that and make the cancer more complicated. Furthermore, for a polycomb repressive complex is the opposite of a super enhancer. All that happens is PRC2 will add a methyl to a histone, which will tag them for repression. PRC1 will find the tags and compact those regions of chromatin to prevent gene activation. So it's making heterochromatin. And this is used in development to silence these developmental genes and maintain the cell's identity. So again, it's what makes your eye cell an eye cell and your liver cell a liver cell. It stops liver genes from being expressed in your eye. And the aberration of this is, again, linked to cancers. So this is the end of my presentation. I hope this was informative for you. Thanks for watching.